Bridge Bio. I'm part of our analytical method development team. And today I'm going to share our work involving the biophysical and molecular characterization of our recombinant AAV species. To start off with my forward-looking statements and disclaimers that all of the work shown is our own interpretation, and I am a shareholder in Bridge Bio. To start off with, I want to give some background on Bridge Bio Pharma. We were founded about 10 years ago and are headquartered in Palo Alto, California. We are a commercial stage biopharmaceutical company that is focused on studying genetic diseases and cancers with clear genetic drivers. rAAV here is a product that recently launched, and we have many other products that are currently in our pipeline. Highlighted in the boxes there, I have our gene therapies. So we have a therapy against Canavan disease that is currently in phase 2 clinical trials, and we have many other programs that are within our preclinical pipeline. More specifically, I'm a part of Bridge Bio Gene Therapy, and we are actually located right here in Raleigh, North Carolina. So we have our lab over on NC State's campus. On site there, it houses our technical development and operation teams, all of our process development work, as well as our analytical method development work. All of our research and development is also done in our lab spaces there. And we have representatives from many other teams that are on site, as well as remote teams that help support our clinical and regulatory needs. So showing here is kind of an overview of our analytical panel. So these are just some of the attributes that we look at within our AAV products. But for the purpose of the talk today, I'm going to be focusing on how we are utilizing long-read next-generation sequencing, or NGS, and where we see that fitting in within our existing analytical panel. And so we see NGS helping to support our identity of our GOI, as well as understanding the purity and seeing what our impurities are within our product, and then also giving us some insight into the strength and potency of it. So all of the NGS work I'm going to be sharing today was utilized with PacBio's AAV pipeline there. So the library preps were done using the recommended AAV workflow from PacBio. They were all sequenced on a PacBio Revio. And all of our analysis that I'm sharing was done in collaboration with FormBio using their AAV analysis. I'm going to share 3 case studies. All of it is using development material that is a self-comp AAV. And these 3 case studies are just going to kind of walk through how we are utilizing NGS here at Bridge Bio. So case 1 is how we see NGS supporting our release panel testing. So currently we utilize AUC, VG over capsid titers to get an idea of this percent full, which we're finding that this term percent full means very different things depending on which assay you're utilizing. So with AUC, a great benefit of it is you do get to look at all of your capsids, so we can identify empty, intermediate, and full capsids. However, it doesn't discriminate the DNA sequences that are present. With vector genome over capsid titer, you do have a little bit more specificity because you can design those primers against your GOI, but you are only looking at a fraction of your DNA. And having the VG and capsid titer, those are two different analyses, so you aren't looking at the exact same sample to get that measurement. We see NGS fitting in here to understand this idea of percent full because NGS can now measure our entire DNA sequence of interest. However, one of the limitations with NGS is once you sequence, you are breaking open those capsids and you are looking at all the DNA that is there, and so you lose a little bit of that resolution that you can get with your capsid species. But what we found interesting is although this percent full means something very different assay to assay, when we look at 2 different lots shown on the graph here, when we made process improvements from lot A to lot B, so looking from that the blue dots onto those pink dots of lot B, is that when we made those process improvements, we're seeing those improvements across all of these different methods. So seeing that percent full increase, whether that be within the capsid or looking at your DNA itself. Another way we see NGS helping with our existing methods is looking at residuals. So here I'm showing our helper plasmid, repcap plasmid, and host cell DNA. And we found that NGS does capture more residuals because you are mapping against the entire plasmid or the entire human genome, whereas those PCR-based methods are only looking, again, at those primers of interest, so you're only getting a small fragment of DNA measured. But what we found is, despite those differences in the two methods, again, when we have process improvements from lot A to lot B, and we see a decrease in those residuals, we're seeing that decrease picked up by NGS and those ddPCR or qPCR-based methods. And looking at that more directly here is showing the full change in that lot A to lot B, and we have that decrease in residuals present. And we were kind of pleasantly surprised with how well the PCR-based methods aligned with our NGS results for that decrease in residuals. So it gets us thinking of this idea of perhaps evaluating NGS for future process development or maybe eventually release testing, because we can perform one assay in NGS as opposed to half a dozen PCR-based assays and get the same results that we need for our product. So case 2 is now diving even deeper into those residuals that we had looked at, and it's seeing how NGS can provide very specific insight into the genome content. So we had regulatory feedback around the presence of a fiber reading frame within our helper plasmid, and it led us to need to evaluate further the levels of fiber present in our sample. So as a first pass to look at this, using ddPCR, we designed primers against the beginning, middle, and end of that fiber reading frame. And we found that those 3 different sets of primers were amplifying the DNA to similar levels. And so an assumption we have is maybe this is indicating to us that we do have that full-length fiber reading frame present because of the equivalent amplification of the DNA at those 3 different sites within the reading frame. However, when we performed NGS, we found that that's not quite the case. So showing here is our NGS results with the fiber reading frame coverage map, as well as looking at the fiber reading frame read lengths. And we found is even though those primers were picking up equal levels, that in the majority of what is mapping to fiber are fragmented DNA. And so the full-length fiber reading frame is only accounting for 0.006% of our total reads in our sample. This is a case where NGS is able to give us a lot more information than just a PCR-based method could give us in looking at this. To take this a step further, because a common thing throughout this whole talk will be, not one assay is going to give us everything that we need to know. We also performed an in vitro transgene expression assay to see if any fiber mRNA was being produced. And so we see in that pink line there, across this MOI set, we have a dose-dependent increase in our GOI expression. We see in the gray line there that we have a constant housekeeping gene expression. And then at the bottom there, I was looking at our fiber mRNA expression, and we saw no fiber mRNA produced in these cells. So collectively, the NGS showed us that there are very little, if at all, of the fiber reading frame present. And then looking further now, we're seeing that there is no fiber mRNA produced. So collectively, being able to combine all these different analytics, it gave us a better understanding of having this fiber reading frame within our helper plasmid. So the last case that I'm going to share is how we are now utilizing NGS to help characterize our intermediate species. So on the left here shows our drug substance. And by AUC, we have 45% full, 25% intermediate, and 22% empties. And through multiple rounds of ultracentrifugation, we were able to get 3 very distinct species as measured by AUC. So we have an empty, intermediate, and full fractions. And as you can see highlighted in the red by the plots there, we were able to get very pure populations of over 90% respectively for the empty, intermediate, and full. And so now we're able to take these 3 distinct species and perform downstream analysis to really understand, especially in the intermediate case, what is being encapsulated. So we wanted to utilize NGS, and so we found intermediate species are often being labeled as this product impurity. but we really want to interrogate, do they have any therapeutic potential? And so on the plot here is our NGS results showing the assigned reads for those 4 different samples of drug substance, full, intermediate, and empty. And we found through all of these different samples, the majority of the reads were mapping in some regard to our AAV genome. And showing that kind of more directly here, comparing our drug substance compared to our intermediate. And we see that the intermediate species did have a higher percentage of helper plasmid, rep-cap plasmid, and host cell DNA, but the majority of the reads were in fact mapping to our AAV genome, with over 92% of those reads mapping there. But we needed to dive into that a lot further to see not only are they mapping, somewhere along the way to our AAV genome, but what are those reads? And we found, so looking at that first category there, the full self-comp AAV, we see within our drug substance, we have over 75% of the AAV reads are mapping to our full self-comp AAV of interest. It was nice to see our full is almost 100% full self-comp AAV. This is also a nice check because the full was collected through or based off of the AUC data, so it is nice to see that the full AUC is also showing up full for our vector genome here. What was interesting is for our intermediates, we do have some level of full self-comp AAV within those intermediate species, but the majority of the AAV reads that we were getting were partial reads. And this is something that we're still currently diving deeper into to understand what are these partial reads. And in the talk before, we saw the amount of heterogeneity that you can have. And so we're now working with Form Bio and taking those actual read files to understand what those partials are, especially within that intermediate population. And now to kind of look more directly, do these intermediate species have any therapeutic potential? We performed an in vitro transgene expression assay, and we performed this 2 different ways. So on the left here, we performed by transducing with an equal vector genome loading, and so this meant a higher volume of intermediate species were added compared to the drug substance. And with that equal vector genome loading, we see that they have a similar level of activity, with the intermediate species maybe slightly more active, potentially because there were no empties or anything else within that sample. But we felt like this is slightly stacking the deck to the intermediates being showing some level of activity by loading based off of that vector genome, because based off the NGS, we knew that there was other DNA present that was not mapping to our full self-comp AAV. So then we performed the same assay, but now instead we transduced with an equal DNA loading, and we see our intermediate species, that line there in the pink, that there is a measurable level of activity coming from our intermediate species. And as we expected based off the NGS, it is lower than our drug substance, but it is a measurable level. So this is something we're excited to understand further and hopefully perform some in vivo studies and really dive deeper into these intermediate species. So our main lessons that we've learned so far by utilizing NGS within our analytical panel is really the best analytical approach is going to be weaving all of these assays together to utilize the strength that they have, but also acknowledge that they do all have their own limitations. And specifically with NGS, we found the analysis is only as good as the sample preparation and as well as the databases that you're feeding into your analysis, so you can really make meaningful conclusions with the data that you get. And the future directions that we see utilizing NGS at BridgeBio is continuing to use it as an orthogonal method to support our existing panel, and then also being able to use it to dive a lot deeper into our understanding. Especially right now, we're looking at that for interrogating intermediate species. And we found this is something that we're going to need to do for every program. So we have seen that intermediates for a self-comp AAV in the same serotype are not equivalent. And so this is something that we'll continually do to really understand for a specific product those intermediates that are involved. The next one here is we want to gain a more complex understanding of the NGS. So as I mentioned, especially those AAV partials, we really need to know kind of what partial they are, what part of our self-comp AAV is in fact being packaged within those intermediates. And ultimately, all of this we're taking and feeding back into our research and development to continue developing new therapies. So with that, I would like to thank the BridgeBio team, also Form Bio for having us here today to give this talk, as well as all the work that they've done in helping us with analyzing our NGS data. Peter Thompson at NC State for helping with our AUC, and Seq Center, so all of the NGS that I shared today, the sequencing was done at Seq Center.